Hi everyone, I'm Cagatay Dursun. I'm PhD candidate at Marquette University, Medical College of Wisconsin, Joint Biomedical Engineering Department. I'll talk about PhenoGeneRanker, a tool for gene prioritization using multiplex heterogeneous networks. Uncovering genotype to phenotype relationship is a fundamental challenge in genomics. Here in this figure, full arrows show the hierarchical framework of the central dogma of molecular biology that depicts the genotype to all the way to phenotype relationship. Effects of genotypic variations can hardly be captured by this cascading information transfer linking the genotypic and phenotypic spaces. We now know that there are nonlinear and interactive relationships, including feedbacks, among all omic compartments. Considering those nonlinearity and interactive relationship would be more effective at capturing the multivariate nature of genotype to phenotype relationship. And gene prioritization is an important step in this endeavor. Generally, high-throughput technologies such as GWAS generate thousands of potential genes for a given phenotype. A short, manageable list is required for biological experiments for further analysis. Network analysis provides a useful representation of the network structure of the molecular basis of complex traits. Moreover, it facilitates the integrative analysis of multi-omics datasets, and this integrative analysis could address the challenge of uncovering genotype to phenotype relationship. And network propagation is a promising network analysis method that have gained popularity recently for gene prioritization, and its premise is that functional related genes tend to be close to each other than to the other genes in the biological networks. Random walk with restart is a particular type of network propagation algorithm. Recently, random walk with restart on multiplex heterogeneous networks has been developed. Here the figure shows this multiplex heterogeneous network. It is multiplex because random walk can utilize multiple layers of gene networks as it is shown in the upper part of the figure. The circle nodes in the upper part show the genes, and one layer could be a co-expression layer, the other layer could be a PPI layer. It is heterogeneous because random walk can utilize phenotype layer, shown on the lower part of the figure. Here those square nodes are phenotype nodes, so this layer could be, for example, a disease similarity layer. One important shortcoming of network propagation methods, and random walk with restart is not an exception for this, is that they are biased toward high degree nodes in the network. So the highly connected nodes in the network are ranked in the top by network propagation methods. We developed PhenoGeneRanker. It can utilize multiplex phenotype network as well as multiplex gene network. So we can have multiple layers on the phenotype side as well, which enables integrating more data sets into the analysis. It also generates empirical p-values along with the network propagation scores to account for the bias inherent in network propagation methods. PhenoGeneRanker is implemented in R and available on this link. We evaluated the performance of PhenoGeneRanker on rice dataset. Rice is one of the primary source food in the world. Cold sensitivity of rice is a major obstacle, especially in the northern hemisphere of the world. We collaborated with our colleagues from Biological Science Department. We were interested in cold tolerance mechanism of rice in early growth stage. We used the experimental data from samples of 360 rice cultivars. Some of those cultivars are known to be cold tolerant, some are sensitive, and some are intermediate. We utilize the phenotype experiment data coming from different temperatures. We use electrolyte leakage level of the shoots and leaves taken from those rice seedlings. EL level is used to measure the cold sensitivity of rice plant. Increase in EL level under cold treatment indicates sensitivity, and decrease in EL level indicates cold tolerance. Also, we utilize the survival rates of these cultivars under different cold treatment temperatures. Here this figure shows how we applied PhenoGeneRanker to rice cold tolerance analysis. Our collaborators implemented a GWAS pipeline which inputs phenotype data and cultivar SNPs data and outputs quantitative trait loci and hence thousands of candidate genes located in these regions. We created undirected and weighted rice multiplex heterogeneous network, which includes those candidate genes coming from GWAS. We use publicly available datasets for genes and phenotype datasets of cultivars. and finally rank the genes and cultivars along with empirical p-values. We conducted a Go-enrichment analysis to assess the gene ranking results. Here, candidate genes are GWAS output genes, and significant genes are the ones that have empirical p-value less than 0.05. We used a multiplex heterogeneous network of 3 gene layer and 3 phenotype layers. For the complete multiplex heterogeneous network, we used gene and cultivar seeds from literature and the phenotype experiment data. As a negative control, we used the bottom-ranked genes for enrichment also, and they were enriched in no-go terms. This is Go term tree in parent-child tree format. Colored boxes show the enriched Go terms. Yellow to red color indicates the increase in significance. Significant candidate genes in the top 200 enriched in cold tolerance related Go terms. All these Go terms are listed in this previously published Sperata paper as rice cold tolerance related terms. So we developed PhenoGeneRanker. It can utilize multiplex phenotype network as well as multiplex gene network. It generates empirical p-values along with gene ranks. Top significant candidate genes were enriched in cold tolerance related Go terms in rice cold tolerance analysis. Multiplex phenotype network capability of PhenoGeneRanker could be effectively utilized where network size is bigger and have complex structures such as drug-gene interaction studies. I would like to thank to my advisors and commemorate my advisor Merishima Oyama, who passed away on February 18, 2020 after a heart attack. I would like to thank to our collaborators, Red Genome Database team and BOSDA lab members. Also, we would like to thank to our funders. Thanks for listening.